It's 5 o'clock. Welcome to all the participants of this month's edition of the Talking Through Our Science webinar. We're really happy that so many people joined us tonight and are interested in this really important topic, and we're excited for the webinar today.
Yeah, indeed, it's one of the topics that registered the most participation, and I think it's because it's indeed very important, and we are talking about alternatives in the use of fetal bovine serum in cell culture. And despite there are known ethical issue and also reproducibility issues, is still very widely used in many laboratories. And as nicely said by the today's speakers, today they will give us some different flavors of FBS media that are available. So then I would like to introduce shortly the speakers, and then we can start.
Yes, so it's my great pleasure to introduce today's speakers. So first of all, we have Karen Bieback. Karen studied biology at the University of Bremen in Germany and received her PhD in 2002 in virology and immunology at the University of Würzburg. Since 2005, she's the head of the Stem Cell and Cell Therapy Research Lab at the Institute of Transfusion Medicine and Immunology in Mannheim and Heidelberg University. And her scientific interest is to bring cells and immunotherapies from the bench to the bedside. So really exciting topic. And then also the establishment of good manufacturing practice protocols for mesenchymal stem cells. This is also a topic that her group is highly active in, and of course in studying replacements for fetal bovine serum, which will be the topic today. Our second speaker, and we are really happy to have him, is Tilo Weber. Tilo graduated with a degree in biology from the University of Würzburg in Germany, and he worked at a biotechnology company that already by that time developed animal-free endotoxin tests. And afterwards, he was an environmental educator in Sicily before joining the Animal Welfare Academy at the German Animal Welfare Federation in 2016. As a scientific officer for alternatives to the use of animals in research, teaching, and education. He organizes hands-on workshops on replacement of FBS, and this project is now shortlisted for the Lush Prize this year. So this is a really important topic, and yeah, we're really, really happy to have the two of you in our webinar. And with that, with no further ado, I would just quickly go over the housekeeping rules. So just to keep the webinar really smoothly, we have the chat function, but please keep in mind if you want to ask a question, and we encourage you to do so, so please make use of this function. Please use the Q&A function. You can see it in the bottom. Depending on the language settings, it's either F&A or Q&A. Just press that button, then the window pops up and you can just submit your questions. And then I would say we are ready to start. And the stage is yours, Karen and Tilo.
So yeah, thank you very much for this nice introduction. And of course, everybody who's here to listen to us. And yeah, I think we start first with some questions to you, so we know a little bit about your background. Ah, nice, basic science and translational science on the top two. Clinical science, organizations, regulatory, and also others. Great, 68 persons voted for it. So thank you. So we know it's getting even more basic science. And then we would like to ask you also two more detailed questions. What you already know about animal-derived laboratory reagents, like how you use them. Serum and antibodies are on the top two. Then there's growth factors coating material, others, and also non-applicable. So assume these are the ones who don't work with animal-derived materials. Well, great results. So then up for the next question. Which are, according to you, the main issues of the use of animal-derived materials? Let's have a look. Ethical concerns on number 1. Then batch-to-batch variability, low reproducibility, presence of undefined unknown factors, then interspecies differences, low translatability to humans, risk of pathogen contaminations, and risk of material cross-contaminations. And others, and one person is not aware of any issues. So I think we can change this today. So thank you very much for answering our questions. So I will now start with my talk, and I'm going to tell you about the issues with animal-derived laboratory reagents, especially about fetal bovine serum and its replacement. So the questions we ask you, we also did ask 551 scientists from 52 countries, and let's see what they said. And it's quite similar of what you said. Most use serum and then second antibodies. And also for the second question is very similar. Most say ethical concerns and batch-to-batch variability. But here you could see that 10% of everybody was not aware, of all scientists here were not aware of any issues. So there's still something to do. So if you're interested in more details about these questions, we asked a total of 19 questions to these scientists, and we published it last month in Engineering and Life Sciences.
Tilo, really quick, we cannot see your slides.
You don't see my slides? Okay. So surprise, surprise. It's serum. What most of these 51 respondents chose. And also, as we said before, similar to you, the ethical concerns and the batch-to-batch variability was the main issue. So as already said, you can see more details on this survey we did on over 550 scientists around the world in our newly published review paper in Engineering and Life Sciences. So why are we here? Why is the use of animal-derived reagents in modern-day science a topic to discuss? Well, one thing is that animal-derived reagents are still routine in cell culture. Especially FBS is almost used everywhere. When you have some cell cultures, you at least heard of it. Even in some methods that are used for replacing animal experiments. And this is also something that has to change, especially because in our opinion, subjecting an animal to harm to obtain substances which can already be replaced is ethically unacceptable. It has to end. Also, as animal-derived reagents, especially FBS, is a natural product, and therefore it is undefined. So it means you don't know what exactly is in it and how much of each of the components. And therefore, there are also many variations between batches of FBS. This will cause a lack of reproducibility, or worse, it might even cause misinterpretations of your research results. Also, as it's undefined and you don't know what exactly is in it, you don't know what substance can be inside and are they may be dangerous. So this is even an extra risk, which can cause the refusal of any products on the market, and in the worst case, even a risk for the patients using these products. So I give a few examples of animal-derived products. Of course, we start with FBS and bovine serum albumin as media supplements, but also cell attachment factors like collagen or matrigel. It comes from cancer cells in mice. Then, of course, antibodies from almost all animals, and enzymes like trypsin or lysosine. But today, we'll focus on fetal bovine serum. So I will give a little warning to everybody watching, because on the following slides, I will show some graphic content and it might be disturbing. So how is FBS being produced? It is a byproduct of the meat and dairy industry. And when a cow is brought to a slaughterhouse, according to the serum industry, it is then at the slaughterhouse, or sometimes during slaughter, accidentally discovered that this cow is pregnant. Which also means that the dams will experience distress and suffering during the transport, because they're just handled like all other animals that are not pregnant. Especially this will be an issue in late periods of gestation. And also pregnant animals should not be transported at all, because it can trigger premature birth or even abort of the fetus, or death of the mother cow if the transport is too long. Then when the cow is slaughtered, the fetus is removed and usually still inside the uterus, and then transported to a separate room that should also be sterile. There, the blood collection starts. A needle is punctured inside the heart of the fetus. And if the heart is still pumping, then this brings out the blood into a separate container. Otherwise, you can use a vacuum pump to retrieve the blood. Then the blood is further processed to obtain the fetal bovine serum. So the fetuses die either while they are in the uterus waiting for the blood collection to start, or during the blood collection by removal of the blood. So also during this whole process, a stunning or killing of the fetus is neither common nor compulsory. And why is this a problem? Well, we can compare how bovine fetuses and human fetuses are treated. If you compare them, you can say that the duration of the pregnancy is very similar, around 9 to 10 months. But humans are born relatively immature and immobile. They can do almost nothing without the help of their parents. Meanwhile, newborn calves need to stand up almost instantly to drink milk or to flee if necessary. Therefore, they are born relatively mature and mobile. However, the sensation of pain is postulated for human fetuses. And therefore, during prenatal surgery, anesthesia is commonly used in humans. But as I said before, there's no anesthesia for bovine fetuses. And there's still no scientific consensus if fetuses are lacking awareness while they're still contained in the uterus. Therefore, there is a high potential for suffering of the fetuses. And it is highly unethical to let the animals die by themselves without applying any anesthesia or mandatory killing method that is certified for this occasion. And basically, all fetuses should be given the benefits of the doubt, especially in this case. So after I shocked now everybody, I can go to the better topics. Then let's talk about replacement of fetal bovine serum. One replacement is human platelet lysate. It is derived from human blood that is collected for the use in medical purposes, but it is expired. And therefore it is ethically acceptable. Of course, in the case if it is from expired donations and from voluntary donors. It is also very strictly controlled, and therefore it has compared to FBS, a very low pathogen and contamination risks. because it is already before planned to be used for medical purposes in humans. Also, for human-derived cells, it is xeno-free. That means it is not a substance from a different species, like from cows used for human cells. Also, for human cells, it is almost universally usable. These are the pros, but there are also some cons. Like FBS, it is also still a natural product. Therefore, it is still undefined. You still don't know exactly what is in it. And also, the availability cannot be upscaled so easily. And it definitely should not compete with medical purposes. Another replacement that can lift many of these issues would be chemically defined media. This means that for these media, you know exactly what is in it and how much it is, and at which concentration. Therefore, it is ethically acceptable and free of pathogens and contaminations, because you know what you put inside of it. And also, of course, it's then xeno-free and by definition defined. Also, it is customized. You can customize it to your own situation with your own cell line and adapt it to your specific experiment. And if you know the composition and you can get the components, you can do it yourself. You can make it in your own lab, like you make other materials for your cells in your own lab. Of course, there are also some cons. For example, if the composition of the chemically defined media is not publicly available, for example, due to proprietary reasons. For example, if the company does not show the composition, it means you don't know if what is in it, so it's still potentially undefined. And also they can change the concentrations or the composition or even add xenogenic substances. Therefore, chemically defined media has to be publicly, the composition has to be publicly known and open source. So everybody knows what is in it and can rely that it is still the same. And if it is changed, you know it is changed. It is changed. Also, there's not yet one universal replacement available. But this is the current moment. However, of course, it also needs additional research. You have to find out where you can get a chemically defined media or how to do it yourself. But we all know that if we don't research before starting an experiment, we waste a lot of time. And nobody put it better than Frank Henry Westheimer, who is quoted to have said, 1 month in the lab saves 1 day in the library. So, after everything is known here, why does change still come so slowly? Where are the challenges here? Well, there's still a lack of knowledge disseminations. As I told before, many use FBS, but not everybody knows what FBS stands for. There's just like 3 letters you put in your cell culture. And even if you know what it stands for, you still don't know what is behind these words, fetal bovine serum, and especially how it is produced and what may it cause to your experiment. That's why we do 2 webinars like this, to change that. Another thing is, sadly, a classic, it's like, that's how it's always been done. Which means, why should we change something? We might be afraid of any innovations because in the end it could work better. And also, changing a system is considered to be inconvenient and consuming many hours and financial resources. And also, test protocols many times still include the use of animal-derived reagents, especially FBS. So this also has to be changed. As long as it's not changed and we have to follow these test protocols, we have to use it. Also, there's a lack of regulations. There's no obligation to use an animal-free reagent. You can still use FBS if you want to, only in very rare cases. But there should be a general obligation that if there's an animal-free reagent available, you should use that and only that. So to give a little outlook, a transition to animal component-free cell culture is in fact achievable. And I think I can best say this with a quote from the newest paper that Karen and me published last week. Even though at first glance, it might appear to be a complex and challenging task, implementing these measures will improve basic research and drug development, and eventually lead to better outcomes for scientific progress and for public health alike, and help to restore faith in the validity of scientific discoveries. So thank you all for listening to me, and sorry for the beginning that you couldn't see my slides. And now, after me, Karen is going to talk about the specific examples and how to replace fetal bovine serum. And in the meantime, in between, we're going to ask you another question. Which are the most useful tools and resources for you to learn about FBS replacements and non-animal reagents? That's very nice. Network activities such as this is the number one. So it's great. Also literature, reviews, and publications. We showed you some, so you can look it up. And congresses and international institutional discussions and lab internal discussions, of course. Then online seminars, videos on demand, congresses, specializations, and dedicated social accounts. So that's great. So, but without further ado, thank you all. And now we'll listen to Karen.
Yeah, thanks a lot. Thanks a lot, Tilo, for this very good presentation. Thanks. First to ask, do you see my slides? Yeah, okay, that's good. Just to be sure, because of course I see them. Yeah, so in the second part, I will now give some examples, some success stories, I would like to say. So alternatives, what to use instead of fetal bovine serum. We already heard about platelet lysate, we heard about chemically defined media, and I will also show some, some ways how to transit from an FBS culture to an FBS-free culture. So first of all, I'd like to highlight again, so FBS is everywhere. So it's not only in basic science and cell biology, physiology experiments, but it's also here in pharmacology, toxicology experiments, and biopharmaceutics. So vaccine production very, very often uses FBS for expanding the cells, and it's also used in cell therapy. So this is the issue I'm working in and why I became interested in replacing FBS. Because, yeah, this is something we want to achieve. So to go for a cell therapy product to be used in patients. Yeah, why is FBS used so regularly and what is it good for? I think everybody knows that it's required for cells to survive. So typically a cell, of course, in the body is associated to neighboring cells. It's also close to the extracellular matrix, and it gets nutrients mainly by the blood plasma, or then, the plasma are present in the tissue. So all these factors then tell the cell to survive, to proliferate. This is mediated by growth factors, but also transport proteins, enzymes, hormones, vitamins, fatty acids, lipids, carbohydrates, amino acids, protease inhibitors, and this is just a short list. Cells need to interact to their neighboring cells. So, and this is controlling the confluence of the cells. This is mediating the interactions, the formation of cell junctions, but it's also controlling the cell density. And these factors also control cell to matrix interactions. So by providing adhesion factors, spreading factors, this is controlled. And of course, in the ultimate composition, cell polarity and architecture is maintained. So if you don't have adhesion factors, if you don't have growth factors, if you don't have vitamins, nutrients in general, then of course the cell cannot function. FBS contains really many undefined factors, and this is also something which contributes to the so-called reproducibility crisis. So more and more people become aware of the fact that data which are published cannot reproduce in other labs. And Monia Baker writes in her Nature article already 2016 that how to respect your cells also affects the reproducibility. So numerous variables can torpedo attempts to replicate cell experiments, from the batch of serum to the shape of growth plates, but there are ways to ensure reliability. So in that paper, she first gives this example. When Alistair Kodabokas tried to engineer muscle fibers in his new laboratory, he saw something strange. The tissue was convulsing. He had been growing fibers from the same mouse-derived clone for years, but these were different. They burned more glucose, contained lower amounts of a protein that promotes faster relaxation and fatigue less readily than those he had grown before in his lab, moved from Dundee, UK, to the University of California, Davis. The difference, he thinks, was due to how cows are raised in the United States. But I think everybody working in the lab is very much aware of the fact that it's not only the difference how the animals are raised, but also batch differences coming from the origin, but also of course coming from the seed of the cows and so on. So the question is, how many of you are fed up by batch testing? I think we, at least in our lab, are, but we still need to continue doing that because we still, at least for certain attempts, need to use FBS still for comparing our alternatives to really convince people that they are better. So this is just an example of one of the batch testings we did, comparing 5 different FBS batches. You can already see that the morphology of the cells is slightly different. Although all look fibroblastoid, their confluence is different, their shape, their confluence is somehow different. And if you have a look below at the proliferation, then you can already appreciate that there are 2 batches which promote proliferation quite well, so achieve 100% confluence of the cells after 160 hours. There's 1 intermediate batch and 2 which initially promote proliferation, but then later on the cells appear to die. And you can already appreciate, if you compare batch number 1 and batch number 3, for instance, that one is promoting some kind of more or less linear proliferation, where the other one is promoting more logarithmic proliferation. So how to compare data produced in one lab with batch number 1, for instance, and in another lab with batch number 3. So it's really important if you want to compare data and if you want to reproduce data, to be also aware of the fact which kind of media I use. Typically you check for the basal media, use DMEM or AlphaMem, but very rarely you get information about the FBS batch which has been used. So in terms of improving reproducibility, I like to ask, eat more chicken than bovine serum. With this tendency of moving or improving reproducibility, there are many alternatives, and there are so many alternatives that there is now already a consensus nomenclature to define those. So here is this picture showing that in the entire box of animal serum is the largest, let's say, but within that you have serum-free medium, which may contain purified animal proteins. You have xeno-free medium, which contain human serum, plasma, or platelet lysate. You have animal component-free, which, for instance, contain plant hydrolysates. You have protein-free, which contain hydrolyzed proteins, native lipids, synthetic peptides, and you have chemically defined, which contain these synthetic peptides, but human recombinant proteins. So here you can already illustrate that there is a transition from FBS containing supplements to chemically defined media, but the number and of course the definition becomes much more defined. I'm sorry. Giving some examples on how to transit from FBS to FBS-free, I will give you some examples. So it's interesting for some researchers, FBS is not at all a topic. So all those researchers working with really primary stem cells, FBS is an entire no-go because they need to control the differentiation of the cells. And due to the variety of factors and the undefined nature of these factors contained in the FBS, they are not able to control their cells anymore. So this holds true for hematopoietic stem cells, where serum-free chemically defined media are already available since 1985. These are the numbers I found. Also protocols for embryonic stem cells already available since a very long time. And of course, also for the induced pluripotent stem cells, there are these serum-free protocols available. And I will now give you an example illustrating why it's so important to have these defined factors. So this is just one publication I picked where people are working from primitive stem cells, trying to differentiate them towards cardiomyocyte progenitor cells. And here you can already see that their culture conditions are highly controlled. So they have first an expansion phase, they have then a differentiation phase, and for this they use xeno-free media. So they also add various compounds. I'm not always able to pronounce them correctly, so therefore I will avoid that, but you can already see that for each of these steps, you will have to add certain factors which either maintain the cells to proliferate so that they can be expanded, or which induce controlled and defined differentiation. And this, as said, cannot be achieved using an entirely undefined composition like FBS. This table here now shows which components these people used. So of course they have their basal medium, which contains all the necessary nutrients, salts, amino acid, vitamins, glucose. It also contains transferrin as an iron carrier. It contains selenite as a scavenger for reactive oxygen species to prevent oxidative damage. There's human serum albumin, so therefore it's xeno-free but not chemically defined, carrier for nutrients. There's an energy source. There are certain amino acids additionally required to support cell growth and proliferation, and there are also vitamins needed to maintain the cell metabolism. So you can already see that there is a list, but it is not as long as you would expect it to be. Yeah, coming to my personal experience, as already said, we are mainly working or aiming to get cell therapies to the patient. So we started, of course, at the laboratory bench and now aim to go to the patient's bedside, which requires that we manufacture the cells under clinical scale GMP conditions. And for that, at least when we started our work here, there was this BSE crisis, and regulatory authorities called to replace FBS to increase the safety for the patients and not to transfer prions by using FBS. So at that time point, we started to replace fetal bovine serum. There are some of the issues were already mentioned by Tilo. So we know that the cells we are working with take up the proteins to a large extent. And of course, this large amount of proteins contained in the cells poses a risk for transmitting prions or viruses. It also poses the risk for transmitting xenoantigens. And of course, you don't want to immunize a patient. At that time point, it was also said that FBS induces genetic and epigenetic instability. And of course, we don't want to transplant transformed cells. Yeah, we want to help the patient, but don't want to induce a tumor in him. And the ill-defined nature, lot-to-lot variability, the poor reproducibility is of course an issue if you want to achieve a standardized cell product, which can be used all over the world. And of course, if you consider large-scale manufacturing, you also have to consider economic issues, sourcing and supply. So our work, and of course also the work of many others, already achieved something. So there is a recent survey amongst GMP manufacturing centers working with mesenchymal stromal cells, and you can already see that a large variety of these centers already use platelet lysate instead of fetal bovine serum, although some of them still rely on FBS. The interesting point is now, although the regulatory authorities in the beginning asked to replace FBS, they now have concerns. They say fetal, at least since the BSE crisis is solved, they now say FBS is really well defined. There is a good safety profile, at least if you use GMP compliant lots, which have been tested very intensely. So they have now some concerns regarding the human replacements, although they are used in patients. They now say if you use them for manufacturing and pool large of these platelet lysates, then you have to implement strategies to improve safety, so like pathogen reduction strategies, for instance. So the ultimate goal is, of course, to also overcome the use of platelet lysate and to go to a chemically defined medium. I would just show you this one slide where you can already appreciate that we worked a long time on replacement of FBS in our MAC cultures. So since 2007, we worked on that. And this is just the summary of all this work. So interestingly, for the cells from bone marrow, which we work with, we favor now the use of human platelet lysate. And for cells from adipose tissue, we in fact favor the use of human serum because the cells proliferate much better. And this is all the people in the lab now really like or hate to work with FBS because the cells proliferate very, very low and they are not so easy to handle. So human supplements make your life much easier, which is good, of course. One point I'd like to highlight is this one. So we also compared cells which were cultured in either fetal bovine serum or two different human preparations. And what we observed is that here you see this kind of volcano plot of gene expression data. To the right, you see the culture in human serum. And to the left, you see the gene expression of cells cultured in fetal bovine serum. And you can already appreciate that fetal bovine serum is inducing the expression of certain genes, whereas the human serum, is not causing a significant change when you compare it to another human component. So from these 34 or 35,000 genes, 12 are differentially expressed when you compare to human conditions, but 90 are differentially expressed in fetal bovine serum. This is a small number, but it's significant because in our hands, at least, it changed also the function of the cells. There were different differentiation, or the cells differentiated to a different extent. They had different adhesion kinetics. Also, when we used them in some experiments where we mimicked the homing of the cells were entirely different. So what we concluded from this experiment is that fetal bovine serum, and we think it's the fetal factors coming within the fetal bovine serum. Yeah, of course, it's an embryonic milieu or fetal milieu, that this is causing dedifferentiation of the cells. And really, the cells change and they behave strange. And this is something I really would raise your awareness. Yeah, I showed this already. So there is already a transition towards platelet, but of course, the goal has to go towards chemically defined. And yeah, where can you find this information? There are databases for that. This is the FBS-free database, which we use to look for chemically defined media. The good thing is that you not only get the list, but they are already evaluated and rated. So you don't need to spend another 15 years on finding an alternative to human platelet lysate, but you can have a look at this and profit from what others have already been doing in the past. Just another example, I think people are aware, or you might be aware of the fact that cell cultures are getting more and more complex, that you want to mimic cell-cell interaction, that you also want to mimic tissue interaction, organ interaction. This is an example of a body on a chip where 4 different tissues have been combined, and the authors claim it's essential that you have a controlled culture environment which promotes all these 4 different cell types, 4 different tissue types. So it's required that you are able to mimic the in vivo situation, that you have chemically defined culture media formulation, even for multi-organ cultures, and then you also consider surface modification to resemble the in situ situation as best as possible. So this is the example of the culture medium they used for these 4-organ biochip. And you can appreciate the list is not so complicated. First of all, I thought it must be really, really complicated to have such a medium which favors 4 different cell types. But as you can appreciate, it's not so complicated and it's not so long, so also not that expensive. Of course, it uses a number of growth factors, which typically are quite expensive, but the more they are used, the more economic they get. Yeah, these are more or less examples if you're working with newly established cells or starting an experiment from the scratch. But as Tilo said, there are already protocols which are defined, for instance, for drug efficacy testing or toxicology testing. And yeah, there should be really a need to replay or to transit from FBS to serum-free chemically defined. And this is just one example of Aline Jarry, recently published. She has been working on changing the culture conditions from this A549 human lung alveolar-like cells, which are used in respiratory research, but also drug efficacy and toxicology testing. So it's also something which is really used in biopharmacy to assure that no products have no toxicity, for instance. And yeah, she really shows how to transit from an FBS culture to an FBS-free culture. And she did it by gradually exchanging the FBS medium with the FBS-free medium, so to adapt the cells very, very slowly to the new conditions. So they're not entirely surprised, but they feel well and get adapted to it. And yeah, here are the data shown compared to the classical medium, which in that fact promoted the proliferation of the cells pretty well. 2 different chemically defined, or at least xeno-free medium, really promote or delayed the proliferation. So they were proliferating much slower, but they had a much, much better dose response curve, which mimics much better the situation which can be found in vivo. So with changing the culture conditions, you can really refine your cultures and yeah, also get a better reflection of what happens in the patients. Yeah, so it's improving your culture. It's not only making life easier, or in that case prolongs the cell culture, but it really changes to an improved testing system. yeah, so to make this long story short, I don't want to repeat everything which has been said. There are many drawbacks of bovine serum. I think that for at least as a scientist, the most important one is this poor reproducibility. This also holds true for human blood-derived components, although they are already an achievement. But chemically defined media should be the ultimate goal we have to go for, because yeah, this is the best control, this is the best standardized. It's also, when you think about applying cells in the clinic, the ultimate goal to achieve. Yet one has to consider that, of course, yeah, cells will lack something, whereas in fetal bovine serum or human components, you have plethora of factors chemically defined will reduce it to the most essential ones required for the cells to survive and also for the cells to function. So this is something you should consider. So any significant change of culture conditions can have an impact on the cellular qualities and needs to be investigated. So you need to validate your cells afterwards. But in many cases, they are better afterwards. So thank you very much for your attention, and yeah, feel free to replace fetal bovine serum. So I will stop.
Thank you very much to the two of you. This was incredible and really exciting to hear about all these alternatives which are already there. So I can see that there are some questions on the Q&A feature. So we can just start, and I can perhaps maybe read the first one to you. So maybe one of you wants to comment how efficient are the alternatives to FBS during the experiments?
I think I showed data, right? So they are very efficient. Yeah, it depends, of course, on your culture conditions. But as I said, so we in our lab, we very much favor the use of, in the moment, humanized culture conditions, but we are also in the progress of improving chemically defined and really cells. Yeah, they grow fast. People like to use it. So when we have to use fetal bovine serum, it's always, oh, do we have to use it? Yeah, so it's good. Yeah. It improves the quality. Yeah, this is important.
We have also another question about the source for growth factor. So what is the source for growth factor required for chemical defined media, and if they can be synthesized? And I think it's linked also to the, if there are, yeah, also about their availability, because there is also a follow-up question if they are available in the US or where this can be found.
Sorry? Yeah, there's a larger variety of recombinant proteins you can order. Yeah, so this is the easiest way to go for chemically defined. If you want to do it on your own, there are chemically defined media which are pre-formulated, so you can buy them, ask the companies, look at the FCS database, whether there are already available protocols, and then if you don't find a commercial one, do your own. Have a look at the papers where you find protocols.
As for example, in our paper, we published a composition for CACO cells, CACO2 cells.
Okay, then we have another question. Is a high throughput screening, such as proteomics of cells, grown using alternatives of FBS?
Yeah, they are. So we try to do that. The issue is that you cannot compare, right? In a proteomic approach, you will not see the bovine proteins, or you cannot detect them, classify them. Yeah, so that's the complicated thing. Of course, you can compare the proteins of your cells, but you cannot see contaminating proteins coming from the FBS. So that's the issue. Of course, there's albumin and so on. So if you want to go for proteomics, you always have to delete that and so on. So that's the complicated thing if you really want to compare, because of course it's xeno, yeah? So if you're working in the human condition and want to compare human proteins. So we try to do that, to really compare the protein content of platelet lysate to FBS, but it was impossible. Yeah, so of course, then we switched to comparing the cells, but still we don't have any information about the contaminating proteins.
Another use that is mentioned is there are alternatives to using FBS for freeze or thawing of primary cells and cell lines.
Yeah, it works perfectly well to replace it. And there are so many protocols now for FBS-free freezing protocols.
Mm-hmm. Going through, and one of these is, does the HPL have batch to batch variability? Have you observed it already, or it doesn't have a significant difference to the FBS?
We were surprised not to see it that much as for FBS. So FBS batches are huge batches. So they are produced from, I don't know how many 100 animals. So they're really, really huge liter batches. And the platelet lysates we use is something like 1 liter in maximum. So it's pooled from 8 donors. And surprisingly, we have seen very little batch differences. They are, of course, but they are not as significant as shown in this FBS batch testing. Yeah, so of course there are something like 5 to 10% difference in the proliferation, but none of them supported, not at all, or something like that. So it's nothing as striking as for FBS batch testing. And if you increase pool size so that ideally you have something like a 5,000 liter pool, then it does not matter for a clinical scale or biopharmaceutical approach, because then you have your one large batch, like the same like for FBS.
Thank you. So also see, so there is some comments to a question that was answered, I think, in a written form about slaughterhouse. I don't know if Tilo, you want to comment on this?
Yes, I can comment on this. So far, there are no informations available if FBS is produced in Germany, but it is well known that FBS is produced in France and Netherlands, and probably also in other EU countries. And yeah, the second part of this question was what is being done against this situation. So my organization, we are working together with the Eurogroup for Animals on the European level to demand a regulation of the FBS production, but ultimately, of course, of a replacement of FBS, so that FBS should not be produced at all and not be demanded, because availables are there and should be used more. So I hope this was sufficient for this question.
Then we have something about money. So are there yet more expensive investment to serum-free or chemical-defined media than using FBS? It is also important.
Well, I can also, first I can say to this, what is more expensive? Like repeating your experiment because you don't know if you can trust the results, or afterwards if you can't repeat it because of the FBS. So this is a problem. And also, well, you know, agriculture and everything is not getting cheaper, especially during these days. So FBS will rise in prices. And at one point, there have to be alternatives. So, and when the demand rises for alternatives, also the price will probably lower because more people will invest on it and produce it.
Yeah, I fully agree. I think there's always this discussion about prices. It's also what we have in the labs. An academic lab never has money and never has money enough for all this basic work. So it's always an argument. But I think if you, as I showed, so the chemically defined media, they are not that complex. So they require 5, 4, if at all, growth factors, which appear to be expensive. But if you think how many liters of medium you can produce with them, it's not that expensive anymore. So chemically defined media, if you buy them commercially, typically they're expensive, but then they are all patent protected, so proprietary. So these media typically are really expensive. So 500 euros for half a liter. This is always where you think, oh, this is not possible in my lab. But once you're able to produce it on your own, it's not expensive anymore. And I think the more it comes, the more pressure there is, the more companies are working on that, the more the price is dropping.
Yes. And for other products you use in the cell culture lab, you also don't say like, yeah, well, instead of highly purified water, just take tap water. It's cheaper.
Yeah, it's the same. Yeah. And people do really sophisticated experiments afterwards, right? Like NGS or something like that, but or single cell experiments. But if you culture your cells in whatever,
yeah. So at the end,
you
you
have
may
to
waste
find
the rest.
amount, right? So you have to start from the beginning with good conditions. Then your experiments are worth it, and people are able to reproduce them. Yeah.
Yeah. I think it's a really good final learning take-home message, and also brings me. So we are at the end of our time, and but it brings me also to the final thoughts that about the next appointment. There will be August culture practice. So I will share later a slide. So this is really strongly related that if you don't trust your culture and what you're doing there, then whatever results you derive from this, yeah, it's all linked together. So it's a very important question and very important matter we should tackle. And yeah, I would really like to thank both Karen and Tilo. This was really informative. And I know there have been probably many other questions, and you can contact us on social media, email, just feel free. And as was already mentioned, the webinar is recorded, so you can also rewatch it or share it, or yeah. So maybe Silke, if you want to wrap it up.
Yes, so as Georgia just mentioned, we will post the recording as soon as possible. If there are any urgent questions left, you can always just email us the same address as you can use for the certificate of attendance, and we will just forward them to our panelists and see what we can do there. All right, then I would like to thank, first of all, of course, Tilo and Karen once more, and then, of course, all the other participants. Thank you for being here, and we are very much looking forward to welcome you to the next webinar. Make sure to register already to save a spot, and then we will see each other in September. Have a nice evening, and
thank you very much.
Bye, everyone.
Bye.
Bye, everyone.
Bye, Tilo.
Bye-bye. Thanks a lot for taking your time.
Thank you.
Thanks.
Thank you.